since we know the outcome for many of these people, how well they did with therapy or without therapy, we can correlate this staining pattern to clinical outcome. Now, if it turns out, it didn't, but if it would turn out that high BCL2 is correlated with poor outcome, that means that we now have a prognosticator for Lyme-Mouse. Does that make sense? And that if we can confirm this, that you can then say, well, we've now confirmed it twice, this can be used as a prognosticator for a patient who comes into the door. For a new patient, we can do that stain and say, this is a bad case of Lyme-Mouse versus this is a case that we can expect to behave less aggressively. And this is just a schematic drawing of how you can take several cuts to these pores. The pores are standing up in the paraffin box. We cut slices through them like this, so that we get little circles. And each of these circles can be stained with different antibodies and can be stored as a digital image in our computer system. And we have now over 300,000 of those images in a database that we use on, I would not say daily, but on a weekly basis. So this slide is just to remind you, to tell you that we're now moving away from gene arrays into a revolutionary technique that has been developed by absolutely brilliant people that allowed us to do gene sequencing of DNA and also RNA on a level that was absolutely unheard of 2 years ago. And so, just to bring this in context, it cost $2 billion, and it took 10 years to sequence the first human genome. Okay, $2 billion. Now, the first human genome is always the hardest, of course. The first thing you do is the hardest. Nevertheless, that's an astounding amount of money. Last year, John Quake, a researcher at Stanford, who started his own company, sequenced his own genome for $40,000 in 4 months. Okay, so we're going from $2 billion 10 years to $40,000 2 months. And the prediction is that within a year, it can be done for about $2,000. Okay. So what we have done, and I'll mention that a little bit later, is we have nothing to do with the development of the technique. That is a completely different field, but we use it, and we now use these machines to do gene expression profiling also. In other words, to determine at which levels the 20,000 genes that the human body has are present in various tumors. So that's what I wanted to say about technology, and I hope I could convey some of my enthusiasm for this technology, because we're really living in dramatic times with respect to cancer research and actually all kinds of research through these revolutions in technology. And you know, it's still expensive. It still costs us about it's about $800 now per sample to do our experiment on. Okay, but that with the funding that we get from the NIH, which is what this first part is about, we can afford that, and as I will show you, we hope to really do this on hundreds of Lyme-Mouse cases, not 10, 20, 30, but hundreds. And I'll explain to you why we believe this is really feasible, and we're actually already doing it. So the rest of the time, I'd like to talk about 4 types of Lyme-Mouse research projects that are very active now in the lab. There's a 5th one that is for which we have done our work, and it's now being finished by a guy, Cheng-Han Lee, who gave a talk here some years ago. You remember him? He's now assistant professor at the University of Vancouver, and he is still very active, involved with the Lyme-Mouse research, but he's setting up his own shop there, and he's taken the rest of that project with him. Maybe he can talk here again sometime about that. So that's the 5th project that we're involved with. So the 4 research projects are here in 3 groups. The first one is the question, is leiomyosarcoma one disease or are there different subtypes? And that's a very expensive study for which I'm very fortunate to have gotten funding by the NIH. As you all know, that funding is now, and you probably know also, funding is very hard to get. So we'll see how long it lasts, but for the next 4 years we have good funding to do this first project. And I'll get into more details about it in a second. The next 3 projects, 2 here and this one here, are supported by the National Leiomyosarcoma Foundation. The first 2 are 2 projects that study the role of macrophages in leiomyosarcoma, and then the last one is a project on a particular protein called ROR2, and I will talk about that in a minute. So let's talk about project 1. So the question is leiomyosarcoma one disease, or are there different types of leiomyosarcoma? Now, as you all know, there's leiomyosarcoma in the uterus, and there's leiomyosarcoma in the rest of the body, right? There's leiomyosarcoma in the female genital tract, as we call it, or in the rest of the body. And so those are 2 types already there, right? They get treated by different clinicians. The classification schemes for it are different. The grading schemes are different. But if you say, okay, well, they're all coming from smooth muscle cells. If we say that is one type, can we find other subdivisions in there? Well, let's first see why that would be an important question to ask, right? Because is it just out of interest, or is there a practical consequence? So let's just hypothetically assume that in this box, each of these circles represents a patient. And now, one patient is one circle, right? And let's now say we're going to try a new drug. We or somebody else, mankind, has found a new potential drug, and we want to see what it does in leiomyosarcoma patients. So we ask 50 patients to be volunteered for this study, and the drug is given to them. Let's say that animal studies were very positive, cell studies were positive. We think this could be a good drug. We give it to these 50 patients, and let's say that only 2 patients respond. Well, if 2 patients respond, it means that 48 of them get treated unnecessarily. And as we all know, each drug has its side effect. You rather not do that. Okay. Is this example clear?
Yeah.
Okay. So, and I like this example because this is something that I actually only realized not all that long ago, maybe 10 years ago, about how this principle works. And this is an important thing. So now let's say that you are able to identify a new subset of cases by whatever technique you use, whether that is looking at a particular gene or a particular protein. But you can say, this patient has it, that patient doesn't have it. This patient belongs in this group, that patient belongs in the other group, right? And that marker, I just call it a marker, that protein, if you will, is indicated by this green dot, okay? So here's a patient with a tumor, here's a patient with a tumor with a particular protein on it. Now, if you know that data, you have a chance of finding that the patients that responded are really overrepresented amongst the patients with the dots. In other words, that only the patients with a dot stand a chance of responding. Okay? Does that make sense at all?
Mm-hmm.
Yeah? I mean, this is hypothetical, right? But it becomes less hypothetical as you have if you have an idea about how this drug could work, and if you have an idea from the gene expression studies at which proteins might be affected by this drug, and then you can look for those proteins in your cases and see, well, this tumor has a very high level of this protein, which is a target of this drug, so let's see how they respond. Now, so if that is the case, right, you see that still 2 patients respond. That is still not many. But if in your next study you would say, I am not going to treat patients unless they have this green dot, and only treat the ones that have the green dot, now your success rate goes up to 50%. Okay? What that means, there's a number of things that means. First of all, drug companies will not toss the drug out, okay? Because they've got a 50% response rate in a selected group of patients. They like that, right? They're not going to say, this is it's a useless drug which costs only 2% of the patient response. Secondly, you've now saved 48 patients exposure to a drug that won't affect them. And they can therefore be treated with other drugs, right? Or they are candidate to be treated with other treatment profiles. You don't have to waste their time and their health by exposing them to this drug. So that's the background of it. And then historically in 2002 we published a paper where we could see that Lyme sarcoma actually lived in two branches on this dendrogram. This is sort of like an evolutionary tree. When tumors are the same, they are together on a branch such as these gysts that are all on the green branch, synovial sarcoma on the blue branch. But you see that the Lyme sarcomas are split out in two groups. And it was the first time that that was shown for Lyme sarcoma, suggesting that there were more than one subtype on a molecular level. In 2000, and this was done on maybe 10 cases or 12 cases or so. In 2010 we published a paper where we could show that on 46 Lyme sarcoma patient samples we could actually find three groups. We call them type 1, 2, 3. And we subsequently published a paper in another journal that looked at the markers that were used, protein markers that can be used to at least identify one of these subtypes. This is still very, very much a work in progress, and it's a long-term progress, and it will take years. But we now have the ability through a technique that was developed in our lab that we can look at paraffin material and enter that into this sequencing machine, right, and get expression data for tumors. And we have now done 60 so far Lyme sarcoma cases. We hope to get all the way up to 300. Bigger is better. More cases is better. We'll be able to analyze more data. And that is...
How long does one analysis take?
Okay, that's a very good question. So I got two ladies in the lab, technicians. They can do, when they work together, they can do 20, they can prepare 20 samples over a period of 2 weeks. Then we're done with our stuff. We've made it ready for sequencing. And then it goes to the sequence facility at Stanford. And that has a turnaround of a month, if all goes well. So we staggered, and so we can do between 30 to 40 samples a month. Yes?
So, a lot of people have sent off saliva samples to